Good afternoon, students. Last we have discussed about the bacterial cell structure. I have shown you the structure of the bacterial cell, okay, and I have told you that the bacterial cell is the sole organism of the kingdom Monera, okay. So what are the features, characteristic features are there in the bacterial cell that depicts the characteristic features of the kingdom Monera, okay. तो उसमें हमने सेल का स्ट्रक्चर जो है वो डिफाइन करके आपको समझा दिया था. Now let me take you through the composition of the different characteristic features of the same bacterial cell. Like first, if we come from the outer to the innermost, the outermost layer, that is known as glycocalyx layer. Glycocalyx layer, okay. This glycocalyx layer in the bacterial cell, the glycocalyx layer is inside made up by two layers. It is composed of two layers. One layer, it is called slime layer, and another layer is there which is called capsule layer, which is known as capsule layer, okay. Now the slime layer, this slime layer is thin and it is elastic type, loosely binded, okay. So this layer is a thin layer, it is loosely binded layer, it is not a firm layer, okay. But the capsule layer, it is a thick layer and it is tough layer, thick and tough layer, fine. This slime layer, it is mainly composed of, it is made up of polysaccharides. It is made up of polysaccharides mainly, and this layer, capsule layer, it is composed or made by polysaccharides plus polypeptides. So it is composed of both polypeptides as well as polysaccharides. Next thing you remember, the slime layer which is present in the bacteria, this layer is non-pathogenic. It is non-pathogenic. It doesn't cause any diseases. It is not a disease causing layer. But capsule layer, it is pathogenic. It is the pathogenic layer. So this is a pathogenic layer, remember. So if any bacteria is having the presence of the capsule layer, that bacteria is able to cause the disorder or diseases. It can be disease causing organism. So that's a pathogenic layer. Another important function of the capsule layer is this capsule layer, it protects the bacteria. The bacteria which is having the capsule layer, it protects the bacteria from the host immune system. So actually this capsule layer, the bacteria which have the capsule layer, those bacteria have the capability that the bacteria, this capsule layer, it hides the bacteria. So bacteria goes hide within this capsule layer, and this capsule layer protects it from the immune system of the host. So that is why the bacterial infections by the capsule layer, caused by the capsule layers, those bacterial infection remains for the longer period of the time, because it helps the bacteria to get hide within this capsule layer, okay. So that is the specialty of this layer present in the bacteria. So remember about the glycocalyx layer. So this glycocalyx layer, it is the secretion, actually it is the secretion of plasma membrane. The plasma membrane of the bacteria, which is made up of lipoproteins. Secretion of the plasma membrane led the formation of the whole glycocalyx layer outside of their body, okay. And this functions like as the glycoproteins which are present in the animal cells. So any foreign substance which comes in contact, that foreign substance that has to go through the glycocalyx layer. So in the bacterial, if there is any bacterial infection, so that for combat the bacterial infection, any drug which is being used to control the bacterial infection, so that drug has to cross the glycocalyx layer first. It has to cross the glycocalyx. So glycocalyx layer is a protective layer for the bacteria. Okay, so that is the composition of the glycocalyx layer. Second thing, after the glycocalyx layer, there is the cell wall in the bacteria. The bacterial cell wall is made up of by peptidoglycans. So they are made up of by peptidoglycans. Peptidoglycans are inside composed of two things. They are composed of two parts. One part, it is the peptide part, peptides, and another part it is made up of by glycans. So peptides and glycans are there present in the peptidoglycans. Peptide part is made up of by various amino acids. That is made up of by several amino acids, and one amino acid bonds with another amino acid, led the formation of the bond which is known as peptide bond. Okay, so that is why this is the peptide bond. Peptide part is made up of amino acids, various amino acids. Glycan part. The glycan part is composed of sugars. It is made up of by glycans which are known as the sugars. So in this glycan part, there are the two important components which are made up of the sugars and the proteinaceous components. They are known as NAG and NAM. In the short form, they are known as the NAG and the NAM. NAG full form is N-acetyl glucosamine, and NAM full form is N-acetyl muramic acid. Okay, so these are the two important glycan sugars, and those sugars are bound to each other. They are binded to each other with the bond. That bond is known as beta 3 glycan bond. Beta 3 glycans. So these are the backbones. It's being known as NAG and the NAM proteins. They are known as the backbone of peptidoglycans. They are the backbone of peptidoglycans. Okay, remember this point. NAG and NAM are the type of the proteinaceous components. They are the backbone of the peptidoglycans. Peptidoglycans are also called as murine. They are also known as murines or muramic acid. Okay, so remember this is the composition of the cell wall of the bacteria. The bacteria is made up of by two parts, peptide part and glycan part, and the backbone of the glycan part is the NAG and the NAM protein. That is the backbone of the peptidoglycans. Remember. Next, after the cell wall, there is a cell membrane. Okay. So the cell membrane of the bacterial cell, it is made up of by lipoproteins like lipids and proteins, like the cell membrane of the animal cells or any other cell, they are also made up of by lipids and proteins. So their membrane is also made up of lipids and lipoproteins. Only in this bacterial cell membrane you have already gone through that there is a modification. There is a modification in the cell membrane of the bacteria. That modification is in presence of mesosomes. Okay, that I have shown you already in the diagram. Mesosomes, how the mesosomes are being formed. So there are two types of mesosomes. One, you have already gone through by that. One is called the central mesosome. Okay, and another is known as peripheral mesosome. Peripheral mesosome, or it is also known as chondroid. It is also known as the chondroid. Okay, functionally the central mesosome, it is mainly responsible for DNA replication. It helps the DNA replication of bacterial cell, as well as it helps the binary fission or division or cell divisioning. Bacterial cell divisioning, it helps in the cell division as well as it helps in the DNA replication. Also it is responsible for cellular respiration. Bacterial cellular respiration. So this is the function of the central mesosome. Like in the same way, the peripheral mesosome is responsible only for cellular respiration. Cellular respiration. So it is not involved in the DNA replication as well as the cell division. It helps only in the cellular respiration. So this is the thing to remember about the mesosome functioning. Okay, so that is the function, mainly function of the outer line of the bacteria, which contains the glycocalyx, cell wall and the cell membrane. Okay, rest you have already gone through inside the cell wall, that inside the bacterial cell there is a nucleoid, okay, which we have already discussed. There is a nucleoid region which is containing the genetic material in the form of DNA and RNA. Okay, and you remember here in this nucleoid region, bacterial cells, they do not have histone proteins. Histone proteins. Okay, so the histone proteins are absent in the bacterial cell, prokaryote cells, but instead of that they have special type of proteins called polyamines. They have the polyamines which helps in the condensation of the genetic material in the central location. Okay, that is the thing to remember. And also after that you have gone through in the bacterial cell plasmids. There are the plasmid DNA. Okay, as I told you, plasmid, there is a double stranded circular DNA present apart from their genomic DNA. This is called plasmid DNA, and in this plasmid DNA there are different genes located in this plasmid DNA. The bacteria which is having the plasmid DNA, inside this plasmid DNA it contains some specific genes. Like there is a one gene which is called F factor gene, or it is also known as fertility gene. This is a special type of the gene. This gene inside the plasmid DNA, the bacteria which contains the gene in the plasmid, those bacteria, they are classified as F positive bacteria. F positive bacteria, it means those bacteria which have the presence of fertility genes, they are considered as donor bacteria. They are considered as donor bacteria. Also, they are known as male type of bacteria. Male type of bacteria, because those bacteria which have the presence of this type of the gene within the plasmid DNA, so the plasmid DNA can be transferred. While during the transfer of the plasmid DNA, this gene can also get transferred. That I will show you how, what is happening. So this fertility gene which is present in the bacteria which contains the plasmid DNA, those bacteria will be called as a positive bacteria, donor bacteria, or male bacteria. Okay, remember this point. Same, the bacteria which does not contain this gene. So there is a bacteria, suppose there is a bacteria, this bacteria, and this bacteria has here plasmid DNA. Fine. This is a plasmid DNA, extra chromosomal DNA, and there is another bacteria, it doesn't have plasmid DNA, it has only genomic DNA. So this will be considered as F positive, this will be considered as F negative. Okay, same, there is a classification that the same bacteria, it is called donor type or it is male type. This will be called as recipient bacteria or female type of the bacteria. Fine, they are classified like that. So always the bacteria which have the presence of plasmid DNAs, those bacteria on their body, they have a presence of pillars. They have a presence of pillars. Okay, which you have already gone in the structure also. Bacteria which do not have the plasmid DNA, they do not have the pillars on their body. If any time the bacteria, positive and negative bacteria, comes in contact each other, if they come in contact each other, fine. So between these two bacteria, when they come close each other, when they come close each other, two bacteria, this bacteria has presence of pillars and it has the presence of plasmid DNA. Okay, this is a genomic DNA. Then one of the pili of this F positive bacteria, it comes in contact with the bacteria which doesn't have, like there is a pili, this pili come in contact with this bacteria, then this pili gets converted into a tube like structure. Here it gets converted into the tube like structure, and the tube is being formed between the two bacteria, but those bacteria are not going fused. Without fusion, this tube combines these bacteria, F positive and F negative bacteria. This is F positive, this is F negative, and this tube, it is known as conjugation tube. It is known as conjugation tube. With the help of this conjugation tube, what happens? This plasmid DNA led the formation of the copy of the same plasmid DNA, and from that certain genes are being transferred. So the genes now get transferred from this plasmid towards this, through this conjugation tube towards this bacteria, and this bacteria also develops the plasmid DNA. So the genes one by one get transferred from donor bacteria to the recipient bacteria, male bacteria to the female bacteria. Then the plasmid is being also developed in this bacteria. With that plasmid, there was a fertility gene. There was the fertility gene, F gene. This gene also gets transferred and gets transferred into this bacteria. So accordingly, when this plasmid get transferred, this gene get transferred, fertility gene, from this bacteria to this bacteria. So the bacteria F negative is, this bacteria gets converted into the F positive now. This bacteria will develop the plasmid DNA also, and this bacteria will develop the pillars on its body as well. So this bacteria got converted from F negative to F positive bacteria. This is a type of changing of the sex in the bacteria, that bacteria was in the one form first, it was a female type. When it develops the fertility gene inside, then it got transferred from female to male type, F negative, from the F negative to the F negative positive type of the bacteria. So this process of changing of the sex within the bacteria, that phenomena is known as sexduction. It is known as sexduction in the bacteria. So bacteria also have been classified based on this kind of the phenomena. Okay, so remember, this is the how the plasmid is important in certain bacteria. With the help of the plasmid, those bacteria become specialized. They become specialized. Okay, so that is about the plasmid. One gene, that this gene can occur, can do the transfer of this in this way and can change the sex of the bacteria. In the same manner, within this plasmid DNA, there are different genes. There is a one gene, another gene called R plasmid gene. Okay, R represents here, it's a resistant gene. It is the resistant gene. Inside the plasmid, there is a one gene which is resistant to certain drugs. It is resistant gene to certain, it has a resistance. This gene has a resistance against certain antibiotics. Okay, so there are some antibiotics which tries to kill the bacteria, but there are the bacteria which have the plasmid inside. Those plasmids have the presence of R plasmid genes. Those genes are inculcating, giving the nature of the defense of the bacteria to fight against these antibiotics. So those antibiotics will not work on those bacteria because they have already natural resistance against these antibiotics. Okay, so like that, there are the different types of the genes which are present in this plasmid DNA, and with the help of those genes, this plasmid kind of bacteria which having the plasmid becomes the specialized type of the bacteria. Okay, remember, that is the function of the plasmid DNA. So after the plasmid, you have already gone through that within the bacterial cell. Within the bacterial cell, there is no cellular organelle. There is no membrane bounded cell organelles. Okay, that you already know, that I have already discussed about it. Apart from that, bacteria have presence of inclusion bodies. They have inclusion bodies. Inclusion bodies are, these are membraneless. Remember, these are also membraneless bodies. Okay, and these inclusion bodies are considered as non-living. These are non-living food storage bodies. Food storage body. So the bacteria stores the food inside their body in the cytoplasmic region, that is in the form of the non-living substance. All those non-living food storage bodies are together known as inclusion bodies. Okay, so different types of inclusion bodies like glycogen. Bacteria store the food in the form of glycogen, in the form of volutin granules, that is the phosphate granule. Okay, and in the form of fat globules. These are all considered as the inclusion bodies. Okay, apart from that, I told you only bacteria has only one organelle. That is, only one organelle is present. That is ribosome, which is 70S type. which is the 70S type of the ribosome. Okay, so rest of the things I have already discussed with you. Apart from that, bacteria have flagellas, bacteria have presence of the polyribosome structures, they have ribosomes, they have enzymes inside the body cell. So the rest of the things I have already discussed with you. Okay, so this is the composition of the complete of structure of the bacterial cell, which represents the completely kingdom Monera. Okay, so that is about the whole thing to discuss about the bacterial cell, which represents the kingdom Monera. Thank you so much. Thank you.